that have been conducted by the Pact CIMAC CIDC network. We're very happy to have you all join us today. This is an educational webinar. We wish to promote kind of all of the work that's been done for the Pact CIMAC CIDC network and all the work validating and harmonizing the assays that we have. The hope would be that we would then be able to promote the use of these assays out into the broader community within Immuno Oncology. So I'll be kicking off today. For those of you who don't know me, my name is Stacey Adam. I'm an Associate Vice President at the Foundation for the National Institutes of Health and have been working to oversee the Pact project for 4+ years now. And so with no further ado, welcome again to this webinar, and we'll go ahead and get started. I'll start with a brief overview, then introduce our speakers, and each of our speakers will go through the work that's been done on our Tier 1 assays, plus some highlights of a few of our Tier 2 assays. So, welcome again. So, for perspective, for the quick overview of the Pact CIMAC CIDC network and its purpose. The purpose is to provide a systematic approach to immune and related oncology biomarker investigation within immuno-oncology trials by supporting development of standardized biomarkers and validated and harmonized assays. Now today, in addition to a number of goals that the initiative seeks to achieve, we'll be focusing primarily on the first two that I've highlighted here on this slide today, which is to select a set of basic biomarkers or Tier 1s, what we consider our assays and then the biomarkers resulting from them for a uniform clinical application, and to basically deploy these across a core network of laboratories to conduct, validate, and standardize these biomarker assays. That will be the focus of this particular webinar, but as you can see, our goals stretch far beyond this for Pact, and truly the ultimate goal is to deploy these assays in the broadest fashion as possible, collect as much data from immuno-oncology trials as we can to really start to build an evidence repository that we can use for further validation of these assays, further kind of use of these assays for precision medicine, and really honing in on being able to target immuno-oncology treatments to the right patients. The CIMAC CIDC Pact network, as you can see here, consists of a number of different pieces interacting. The clinical trials, which are often sponsored by NCI, industry, or academia, come in through two routes, either through NCI, through the NCI clinical trial networks, such as the NCTN, the ETCTN, etc., or through the Pact mechanism, which could include IISs, other industry type trials, or even academic trials that come in through that fashion. These trials are brought in, and while we do not support the full trial itself, we do support additional correlative assays being conducted with the trials that choose to partner with us. These trials are reviewed, they are brought in, and then the additional correlative assays are conducted at one of the 4 CIMACs, the Centers for Immune Monitoring and Analysis, the Cancer Immune Monitoring and Analysis Centers. And these are at Dana-Farber, Mount Sinai, Stanford, and MD Anderson. Once the assays have been conducted at these CIMACs, the data goes into a central repository, the Cancer Immunologic Data Commons, which is housed at Dana-Farber. And as you can see, this database not only houses the data from the assays that are conducted, but also the clinical trial data that is matched up to these particular assays. Now, in addition to these academic partners and the funding that was received from the NCI, there was funding received from the private sector. So approximately $60 million put up by NCI and another $60 million put up by the Pact partners, who are the 12 company partners that you see there. This was done as $5 million over 5 years from each partner. Now, as I said, today, what we are focusing on is the assays themselves and the biomarkers developed from those assays. And so as you can see here, this lists the Tier 1, the Tier 2, and the Tier 3 assays. Again, our focus for today will be on the Tier 1s. And as you can see, we have genomic assays, so Wes, RNA-seq, and a number of proteomic assays, including multiplex IHC and O-Link, which we'll be focusing today, and then also CyTOF, also a proteomic assay, but done more in blood-based and single-cell assays. And then finally, we'll briefly touch upon microbiome. Now, most of the assays that I've just named are the ones that were fully validated and then harmonized where necessary across the various CIMACs. As you can see to the far right, we do have additional assays that we are working on to fully develop. We consider these tier 2 and tier 3. This means they've been validated, but not necessarily harmonized across all the different CIMACs because they may be conducted at only a single CIMAC at this time. And for that perspective, this is just the assay platforms and at which CIMACs they are conducted. As you can see, for some assays such as CyTOF, they're conducted across all 4, and for some, maybe just 2 or 3. And then for even more than some like O-Link, which are in our tier 1, conducted at a single site. So again, validated and harmonized when necessary. And again, you can see from the bottom that each of these CIMACs do have a number of other assays that they can conduct, and we do that on considering on a trial-by-trial basis where the biological need to ascertain something allows for those assays to be conducted. Currently, we have a number of trials already choosing to partner with us. This is at last count 33. I think we now have a little bit over that, probably closer to 35. As you can see, they span a wide variety of cancers and a wide variety of treatment types. We are still looking for further trials to partner with us, and we'll talk a little bit more about that at the end of the webinar. But without further ado, I'd like to turn this over to our presenters because the main focus of this will be the assays. So today we have with us Dr. Ignacio Westuba, Chair and Professor of the Department of Translational Medicine, Molecular Pathology at the University of Texas MD Anderson Center. Kathy Wu, the Chief Division of Stem Cell Transplant and Cellular Therapies, Professor of Medicine at Harvard School of Medicine. Holden Maker, Dr. Holden Maker, Professor Microbiology and Immunology, Stanford University, Director of Human Immune Monitoring Center and Director of Services for Centers of Enabling Technology. Dr. Sasha Nachik, the Associate Director of Human Immune Monitoring Center, Mount Sinai, Associate Professor of Hematology and Medical Oncology and Oncologic Sciences at Icahn School of Medicine. So I welcome all 4 speakers, and to start us off, I will turn this over to Dr. Ignacio Westuba. Thanks, Ignacio. You can take it from here.
Good, thank you very much. Thank you, Stacy, and thank you everyone for joining today in this webinar. My presentation is to highlight a protocol that was developed for the CIMAC studies called Umbrella Protocol for the right collection, handling, transportation, and processing in biorepository of samples for immune profiling studies as part of the CIMAC CIDC network assays available. So this is an effort that was actually performed for a large group of people with strong support from NCI leadership that oversee our program. So before I start, I would like to let you know that this document is available with a series of appendices in our CIMAC network website. So you click, use this link, you can see all the detail. Today, I'm going to provide just an overview of the Umbrella Protocol. So before, I would like to use this slide that was shared by one of our CIMAC PIs, Dr. Ignatic, about what we intend to do when we perform analysis on clinical trials associated with immunotherapy. We want to be comprehensive in our approach, hopefully longitudinal in collection of sample for analysis and patient-centric. It's hard to get this in large multi-institutional clinical trials, but we always need to try to be as comprehensive, longitudinal, patient-centric as possible. And also, and you will hear from the expert, there is a large variety of assays, immunology-based or genomic-based, that are excuse me, available for tumor, normal tissues, and patients' samples profiling. I'm not going to go in detail. And Stacy already showed that we have 3 categories of assays that we plan as a network. The tier 1 and the tier 2 are listed in this busy table. Apologize for that, but you can see that there are genomic and immune related biomarkers. And the tier 1, she mentioned already, whole exome sequencing, RNA sequencing, and as an alternative, nanostring analysis for gene expression of immune-related signatures. On imaging on tissue, we have immunohistochemistry, singleplex, and multiplex immunohistochemistry or multiplex immunofluorescence. And proteomics and seromics, we have CyTOF and Olink. And we have a combination of other options as tier 2, and I would like to highlight TCR sequencing that is becoming very much used by different assays and also cell-free DNA. But we have opportunity to do microbiome of stools, expand on the serology, and also expand on multiplex immune profiling imaging of tissue with higher level of multiplex assay, like maybe or others in the future. And all these assays, as you well know, require the right specimen, and also require that the specimen is handled in a way to minimize the pre-analytical variables. And the specimens are tissue, tumor, adjacent, normal, or normal in case of interest to study immune toxicity. Tissue can be fresh or fixed in formalin and for preservation. Also, we have blood derivatives like plasma, peripheral blood mononuclear cells, and then we have other fluids like CSF, cerebrospinal fluid, for example. And also we have some special samples that can come from the bone marrow, like bone marrow aspirate or tissue. And I already mentioned the opportunity to do stools microbiome. So the scope of the umbrella protocol was to establish a consistent process for the sites collecting specimens and the biorepositories involved in the CIMAC study to collect and process tissues, stool, blood, and other fluids, specimens for immune monitoring and profiling analysis to be performed by our network. We also developed standard operation procedures, SOP, that define option for collection, handling, processing, and freezing protocol of specimens, including, as I mentioned, different specimens. It's important to say that these protocols have different options sometimes to accommodate to the reality of clinical research. And the focus is to have the right sample for the right assay, minimizing pre-analytical variables. Very quickly, I'm going to go through the list of, you can see specimen types, some important issues of the collections, some important aspects of transportation that may affect the quality of the specimen. So initially here we have tissue specimen, fixed in formalin or fresh, that are collected for the trial. We call this de novo specimens, or use of archival tissue, usually formalin fixed, paraffin embedded, that was collected as part of the diagnosis of a tumor and a standard of care. In the sample that we want to collect for the study from the tumor, if there are corneal biopsy, for example, we ask for at least 2 cores, preferably 4, or for endoscopy biopsies. When you have surgical resection, you have more tissue, right, available. It's important that this tissue, if it's fixed in formalin and for no longer than 24 hours. If not, it needs to be transferred to another liquid like ethanol for transportations to a biorepository for final processing and make a paraffin block. You have paraffin block archival material, so you can ship hopefully the block, which is a challenge, but if not, we have the option of unstained slide that's been core punches and also scrolls from the block. And the fixed tissue can be sent at room temperature, but the fresh frozen, as you know, has to be sent frozen. Regarding blood, we have different options. We try to collect blood at multiple time points, easy to obtain, right? We have the opportunity to get whole blood that can be helpful for isolated plasma, PBMCs, And from there, there are derivatives like DNA for germline and TCR sequencing. So we have the check tubes that are, very good for stabilizing cell-free DNA in the plasma, and also the PBMCs can be used for germline on the same specimen. And then we have these special K2 purple top tubes that have EDTA that can be used for different purposes. A small draw for germline only, if that's missing, or TCR sequencing, or larger draw for different purposes. So as you can see here, there are different conditions to be shipped, and most of these can be shipped using cold conditions. So the next one is the special collections. I'm not going to go in detail, but I would like to highlight the bone marrow, CSF, for stool specimen. And for stool specimens, we need to ship all these samples frozen. And finally, the samples need to come with data. We need to know about collection times, collection processing, and the protocol. You will see that, for example, for tissue, there are 3 or 4 time points that are important to be collected. The number, the clinical context of the specimens, so we can actually incorporate that in the analysis. Briefly, some details on the tissue collection. I mentioned here that for endoscopy biopsies or core needle biopsies, we like to have 4 cores, and there is a sequence that 1 and 3 could be allocated to fixation in formalin and make a block, 2 and 4 fresh frozen. And here we have already planned, the allocation of those samples for different assays like tissue-based imaging analysis or genomic analysis by extracting DNA and RNA from either the fresh or the frozen if the fresh is not available. It's important that we always need to have an H&E, an histology slide for quality control. If not, an imaging could be important, could be also useful. It's important to, as I mentioned before, to control the quality of the tissue specimen before fixation or freezing. And here we have different notes that allow us to actually get specific information on this. I would like to indicate that we consider the fine needle aspiration, which is cytology specimens, is not acceptable. And for our analysis so far, they do not replace tissue specimen. This is an overview of the option with tissue, surgical resection, core needle biopsy, endoscopy samples that can be fresh frozen or fixed in formalin or in a block, a tissue block, and from the tissue block also section for different purposes are also acceptable. I'm not going to go in details on this table, but we also in the umbrella protocol, we have detailed considerations on shipping specimen to a biobank and also from the biobank to our CIMAC laboratories. And here, for example, in tissue, you have, when it's fixed, option 1 or 2. Basically, option 1 is you have the tissue in formalin liquid, so you need to send it to a biorepository, a bank, which is going to process the tissue and make a block, or you can send a block. And those actually have different requirements in terms of shipment conditions. So for blood, I'm not going to go too much in detail, but as already mentioned, we have the green top tube for different purposes that have whole blood. Then this streak tubes for cell-free DNA mostly, and then the purple top tubes for germline or TCR sequencing with smaller volume than the green top tubes. There is a series of important considerations for blood collection, handling, and shipment, and I'm not going to go through them. Some of them are listed here, but you can find all those details in the protocol. Then also we have a special information for the biorepositories that are going to receive this from the clinical trial site on how much aliquots can be prepared for different assays that we have considered. So the samples of the blood samples. Most of them can be cheap at ambient temperature, unless you have developed derivatives from this blood, like DNA or RNA. So, however, the stools specimens, depending on the kit that we're using, have to be submitted having a cold chain or an ambient, if they have the right stabilizer for the material of the specimen. So I would like to end by showing you again our link to find the umbrella protocol, and thank you for your attention and all the people who participating in putting this important protocol together. Thank you very much. Going back to you, Stacy.
Thanks, Ignacio. And now to introduce Dr. Kathy Wu to talk a little bit about our whole exome and other genomic-based assays. Kathy?
Thank you for that introduction. And I'm really pleased to be here and share with you some of our efforts across the network to harmonize our Tier 1 assays. And so I'm going to speak on behalf of the network in terms of our genomic assay harmonization. And so as I start, these are my disclosures, but I want to just say that this has been really a large-scale effort, and it's just been such a pleasure to work not only with the NCI, but colleagues at MD Anderson, and also within our Dana-Farber, CIMAC, and CIDC network, as well as our close colleagues over at the Broad Institute. And I think I think we would all agree that it's really been a major change in our toolkit to be able to have genomic assays available. This is already a very outdated slide, but I think we would all agree that we've had DNA and RNA sequencing available to us for just about a decade now. And I think from the very first moment, this is from efforts through the TCGA, where we started to characterize the whole spectrum of malignancies across the board. It became quite apparent that we could learn so much in terms of the mutational events, the pathways that were involved, that there were similarities, but there was also really quite a bit of differences amongst these different tumors. And so this is just a brief scan of somatic mutation frequency, and you can see very well that there's some tumors that have high mutation load, such as the carcinogen-driven tumors, way on the right-hand side, and then way on the left-hand side, you have blood malignancies and pediatric malignancies. Again, this is at the time seemed like a huge amount of data, and just imagine in the 7-8 years since, so much more has been done. So I think with regards to immuno-oncology, these are critical tools as we start to think about biomarkers, because they allow us to systematically look across the genome for clues as to sources of therapeutic response and resistance. And so if we think about what we can learn from exome sequencing, these could be on the realm of looking at mutational burden. These could be in the realm of thinking about antigenic sources, such as neoantigens. When we think about RNA sequencing data, does this phenotypically tell us, are there affected pathways that are associated with response or resistance? And can we even have evidence of footprints of immune cell involvement within the tissue? So for example, through RNA sequencing data, could we see evidence of immune cell infiltration by the basis of detection of their profiles? And could that, for example, relate to response or lack of response? And finally, when we start to think about adaptive immune cell populations as T cells or even B cells, can we start to look at immune repertoire, and how does that become altered with therapy? And so as we kind of started to think about this from a network perspective and having these genomic assays available across multiple centers, obviously we were confronted with the question of concordance of our measurements across different sequencing centers, whether it's at the DNA or the RNA level, and looking at the questions of, these very technical questions of whether or not there'd be variation within a site or between sites. And could we actually start to look at real world samples? And so, for example, working closely in hand with both folks at the NCI and also at MD Anderson, we started to think about the HapMap samples. I'll come back to that. And also having available real life patient tumor specimens that we could use to try to benchmark and evaluate this. And I'm happy to report that our work in this area was recently published. Okay, so what was the design? How did we approach this? So really we did two flavors of experiments. The first was to use HapMap pools. So these are very well genomically characterized cell lines. And we basically had mixes of known composition in two different assortments. And these were standardized pools at some level, ground truth data, so that we knew what were the proportions of cells within a pool of DNA that comprised these various subpopulations, sublines. And so, as you just heard from Ignacio, Professor Wistuba, what was important to us was to have centralized pool preparation, centralized DNA extraction. And we also tested, these were freshly frozen, and with the central extraction that was done as replicates, this was then distributed across 3 different sequencing centers. And then exome sequencing was performed, and then we were able to put all the data through a centralized pipeline organized by the CIDC, and we performed not only centralized analysis, but also analysis at the individual centers, and we were able to do those comparisons. A second flavor of experiments was actually to go to actual patient samples, and so the group at MD Anderson, the MD Anderson CIMAC was able to identify 8 tumor specimens from patients with non-small cell lung cancer. And these were specimens that were selected because there was sufficient tissue to compare not only the freshly frozen sample, but also paired FFPE. Now, obviously from the same sample, these were biopsied at different points in the tissue, but it allowed us to be able to start to make those type of comparisons. Again, this was centrally collected, centrally extracted, replicates were generated. Because we were looking at DNA analysis, there was also matched PBMCs as a germline control. DNA was also extracted centrally, and then again distributed across the centers. This tissue was then used for exome sequencing and RNA sequencing, and again through pipelines for data analysis through CIDC or center-specific analysis on for the exomes, and CIDC for RNA sequencing. I'll run through these relatively quickly because this is published data, but just to say at the level of the DNA analysis, all 3 centers were able to capture largely concordant exomes. And then as we started to look at number of mutations, whether in the fresh frozen or in the FFPE, you can see visually that there was a great deal of concordance as we were going through the different patient samples. So that was actually really great. And then as we started to look at the consistency of high mutation call, what came through was that it was really the biologic groupings that came out as opposed to the manner in which the samples were collected. So for example, in the HapMap samples, you could see that the replicates are clustering together, and also again across the different patient samples, whether it was fresh frozen or FFPE, all the patient samples seemed to cluster together based on their molecular features. So that was actually super reassuring. And again, looking at the measures of precision and recall across the HapMap cell line pools, these were all at very high levels. So again, very reassuring that despite different centers, different chemistries, sequencing at the different centers in terms of the analysis, the data kind of came through very nicely. Same thing for the RNA sequencing. We learned a lot through putting these samples through, and we're able to come up with some metrics in terms of QC criteria. So the measure of DV200 greater than 24, whether for RNA-seq or nanostrings seemed to be a really nice measure for being able to generate good data. Med-tin greater than 30 was another criteria. And in terms of downstream analyses using these data that were thus filtered, we were able to have great deal of consistency. And again, I think this is a little bit of a complicated figure to look at, but the groupings of the patients are kind of shaded in this line here. And what this is conveying is, again, the samples all derived, replicates, different centers for the same patient all seem to come together. Now, we were able to have a little fun and pull together the exome and the RNA sequencing data because that's a lot of what we want to know when we go toward our clinical trial analyses. And so what this is trying to say is that as we started to look at what were the prevalent mutations in these non-small cell lung cancer patients, reassuringly, these were the known drivers that seemed to come up. And I think that if you kind of look across each of these rows, it's telling you that there's consistency of detection across the three centers in the green, red, and blue for detection of these highly prevalent mutations across different types of mutations, whether they're missense, splice site, nonsense, or so on. Also, as we started to look at HLA typing, whether it was from fresh frozen or FFPE or between the exomes and the RNA sequencing using different tools, there was a high degree of consistency. And also looking at neoantigen load, this was also, there was a lot of high concordance there. And so I think that's where things stand in terms of our harmonization. We were very satisfied and really pleased that we were able to generate a centralized pipeline that would allow us to ingest both DNA and RNA sequencing data. I want to give you an example of where we've begun to apply our pipelines to the analysis of data. And this was a study that we reported out earlier this year. It falls on the heels of the clinical report of using immune checkpoint blockade for the treatment of relapse hematologic malignancies after transplant that Matt Davids reported out earlier the year before. This was an ETCTN trial. When Livius Penter and my group came upon this, he was really interested in focusing on the myeloid malignancies. And we were able to access the capabilities of the CIMAC network. I think shortly you're going to hear from my other colleagues of looking through some of the other platforms, but certainly in this study, we were able to access the genomic assays as well as some of the Olink assays, and I'll tell you a little bit about this. So by way of background, transplant is a very informative platform for immunotherapy because for sure there is reconstitution of normal immune elements. And it's been asked, how can we improve the outcomes of patients going through transplant who have the risk of relapse? Can we, for example, give immune checkpoint blockade, which has been such an important immuno-oncology agent across malignancies? And could this, for example, help with the problem of relapse after hematopoietic stem cell transplant? And if we do so, is there a risk of more toxicity? Can we actually enhance the impact of donor immune reconstitution? And so certainly with the clinical trial that Matt Davids set up where patients were given either ipilimumab or nivolumab, could we, for example, identify different patient cohorts, those that were responders or transient responders or complete responders? And could we, for example, take a look at the local effects of these immune checkpoint blockade agents at disease sites following transplant? Could we understand the molecular features of response? And could we also look at the systemic effects that are detectable in blood? I won't go through all of this today because, again, this is published, but I just want to take you through some of this. On the ETCTN trial, there were 71 enrolled patients, and you can see that some of them got IPI in the yellow, some got Nivo in the red. The large majority of the patients enrolled did have myeloid malignancy. So we opted to focus on the gold group, the myeloid malignancies with IPI. And what was really very exciting was that there was actually very nice sample collection on this trial. If you focus your attention on the black diamonds, these were disease-specific biopsies. If you focus your attention on the red circles, these were blood samples that were collected. And these patients could be segregated on these different response groups. So in the dark blue, responders, in the light blue, transient responders, and in the red, the non-responders. And I think what you can see here way on the right is that we did confront the challenge of having material come from many different disease sites, whether in bone marrow or skin or extramedullary tissue. And so one of the questions we asked ourselves is, if we have all this tissue, can we actually start to scan, and can we use transcriptome level changes to give us some sort of understanding of what is happening in responders versus non-responders? And I think just to take you back here, what was nice is that in many cases, we had paired pre and post samples. So I think that's important. Again, I want to emphasize that this was a heterogeneous cohort. There were lymphoid and myeloid malignancy patients. There was a number of patients treated with IPI. There were a subset that had matched baseline and on-treatment biopsy. But I think in terms of data generation, and this was RNA sequencing performed on FFPE samples across a variety of tissues by our CIDC pipelines, we could see that there was a nice data that was generated. There was only very few samples that actually dropped out based on our QC metrics. And so we were able to go forth and focusing first on the matched cohort. So across the top, there was a handful of patients that we had matched pre versus post IPI. And looking at their transcriptome profiles, what jumped out at us was actually there was, it seemed to be an upregulation of genes attributed to lymphocytes. And this was also evident on pathway analysis. And when we started to add in the unmatched biopsies by PCA plot, you could see that the responders seem to segregate together, the non-responders seem to segregate together. And this seemed to support this idea of immune infiltration, which we further confirmed when we used different RNA sequencing tools, including CyberSort and Trust, that allows us to look at the RNA sequencing data. And we could in fact see that those responders with ipilimumab seem to have greater infiltration of T cells following ipilimumab consistently across these different, two different tools. So that was actually exciting. One more piece of data is that we started to look at the T cell clones, and overall there was actually very little detectable T cell turnover. So again, focusing on abundance at baseline versus abundance after checkpoint, you can see in the color that there are some contracting and expanding clonotypes, although not dramatic. But if you start to focus on those that are responders and non-responders, the dynamic TCRs seem to be more evident in the responders, suggesting that there's increasing tissue trafficking with checkpoint blockade. And what was exciting is that we were able to work closely with Mount Sinai CIMAC and start to look at the serologic profiling and looking at soluble factors. And what we identified in those responders here in the dark blue compared to the non-responders is actually very dramatic increased detection of these many chemokines and cytokines, suggesting again that ipilimumab response is associated with increased lymphocyte activation and trafficking. So again, super excited to apply these type of assays to the different clinical trials that we're working on. In the last two slides, I just wanted to mention that our original harmonization was performed using the Illumina baits, and we actually are continuously interested in trying to improve and advance the sensitivity of our analyses. And working together with the Broad platform, we've identified that the twist exomes are actually a nice design that will enable us to actually capture more territory with more reliability. And we've been able to share this kind of new sequencing chemistry together with our partners at MD Anderson so that, again, we have consistency across our platform. I won't go into this in great depth, only to say that we've done an in-depth comparison between our old platform and our new platform, and there's preservation of the good features with added targets that we can detect and at greater depth. And I think this gives you just an example where we are cognizant of genomic regions of interest that are just difficult to sequence, just for example, because of high GC content that results in low coverage and decreased mutation detection using older platforms. And yet with Twist, we're able to improve target design and capture. And so I'll just end by saying that we have completed our tier 1 exome and RNA sequencing harmonization. We are continually seeking to improve our platform, and this is a hand-in-glove effort together with CIDC to match up our analytics. We have been, we're in the middle of using those HapMap samples that I discussed with you before to, again, benchmark our older platform with our new platform so that we have good benchmarking. And as I discussed, we've already been generating exciting data on the ETCTN 9204 trial. I think it's exciting to see like that in a very different setting, blood malignancies and transplant, that we see very similar sorts of insights that have already been identified in solid tumor malignancies, really extending our understanding of immune checkpoint blockade. And we look forward also to cross-comparing in the future to other clinical trials that are looking at combinations of immune checkpoint following stem cell transplantation. So I'll end there, and I am very happy to take any questions.
Thanks, Kathy. And with that, I'm going to introduce Dr. Holden Maker from Stanford, and he's going to talk to us about CyTOF, or cytometry by time of flight. Holden?
Thanks, Stacy. This is an effort which we recently published along with the other standardization protocols from the CIMAC network, and was largely led by Adib Rahman, who until very recently was leading the CyTOF group at Mount Sinai, and also with a lot of input from the Stanford group, including Mina Pichavant, Bita Sahaf, and myself. And so that's what I'm going to be showing you here today. But to put it into a little bit of context, this was actually not the first CyTOF harmonization effort that's been undertaken. It's actually the second, as I was involved in the first one as well. The first one being sponsored by the Bill and Melinda Gates Foundation in order to look at concordance of CyTOF data across 6 different BMGF supported sites. And just as you see here, the details of that, we shared liquid reagents and did manual gating across the different data sets for that effort, which was published in Cytometry in 2018. The new effort through CIMAC took advantage of some of the lessons from that first study and used lyophilized reagents, and also augmented the analysis with some automated platforms. So just to touch briefly on this issue of liquid versus lyophilized reagents, this is something that we noted from that first study of Leipold et al., where these pre-made liquid cocktails were distributed to the different sites. They were shipped at 4 degrees, and the sites used them actually within about a week of generating the cocktails. But nevertheless, this is long enough to see some drag up some additional background staining in certain populations. So you can see here, as compared to cells that were provided pre-stained and frozen, the pattern when the sites stained them with these liquid cocktails is a little bit different, and there's some drag up of the negative population here for CD14 and CD16, or here, in this case, this NK cell population shifts up and over a little bit for CD16 and CD56. So not really, lethal problems, but changes in the patterns with even a relatively short period of time, keeping these liquid cocktails at 4 degrees. Now, in the meantime, the group of Henrik Mai in Berlin published a protocol showing that you can freeze these antibody cocktails, particularly at -80 or below, and retain very nice discrete staining, very similar to the fresh cocktail if you do that. But obviously, if you store at 4 degrees or room temperature, that staining degrades considerably. So the other way to do this is with frozen antibody cocktails, but obviously lyophilized cocktails are, sort of the best standard in terms of preserving the staining qualities of the reagents. So that's what we did in the CIMAC study, which I'll now move to. Again, supported by Pact and the CIMAC network, and led by the group at Sinai and Stanford. So we had this lyophilized CyTOF panel that Mount Sinai had generated in collaboration with the HIP-C network. It's a 14 marker panel, which is admittedly a little bit underwhelming in terms of the capabilities of CyTOF, but it's meant to be just a backbone panel of common lineage markers to which you can add in additional markers, which we did for this study. So this was our lyophilized panel. We provided additional liquid antibodies, not as a cocktail, we learned our lesson from the Leipold study, but individual liquid reagents, which were added in for these markers that you see here. Those were provided centrally by Mount Sinai, along with 4 sets of cryopreserved healthy donor PBMCs and a reference control lyophilized PBMC preparation, which was labeled with tantalum to separate it. So this could be spiked in to the other samples and then analyzed separately in order to determine any changes over the period of the run. And then we also had an additional control, which was created by the Dana-Farber, which is a mixture of stimulated and unstimulated PBMCs that were cryopreserved in liquid nitrogen. So the lyophilized control cells have advantages of stability and easy shipping and so on. And they can be used as a spike-in because they're pre-labeled with a heavy metal. These controls over here have the advantage of staining a little bit better with some of the markers because they're not lyophilized, they're actually live cells. And they also have the advantage of having stimulated cells mixed into them, so they'll work better in terms of showing the staining of some of the activation markers in the panel. But they were a separate sample. They were not spiked in to the PBMCs of the healthy donors. Okay, and then as I already alluded to, we also had pre-stained PBMCs that were provided for each of these 4 donors, so we could compare the effect of staining on site as a sort of separate variable. So each site would stain their 4 PBMCs over here, and they would also run a pre-stained tube of those same PBMCs, a separate aliquot of those PBMCs that was pre-stained at Mount Sinai. All right, so lots of setups, lots of controls, 4 sites doing 4 different donors along with these controls. This is then the analysis pipeline that was run. This was also led by Mount Sinai, and it's rather similar to what is done by the Astrolabe system, which we are using as a kind of standard method for doing gating and analysis of CyTOF data in the network. This is just one that was really created by Adib, and so it's doing a similar thing, but it's a sort of a homegrown pipeline. It obviously does all of the required steps in terms of normalization to intensity based beads, eliminating of doublets and separating the beads from the cells, identifying the live CD45 positive events, and CD45 was actually added in a second round of this study because we realized that it was helpful to have a common denominator for all of the immune cell subsets that would help standardize the numbers that came out of each site. And then finally, separating out the reference PBMCs and doing the manual gating in Cytobank, as well as automated gating that I'll show you. All right, so in a first round, I alluded to there being 2 rounds here. There were some issues with a couple of the sites in terms of cell recovery. And I show this really just to be, fully disclosing that not everything works perfectly the first time. And in fact, there is a big dependency on how well the SOP is written and interpretable at each site. And so it turned out that we just had to look at some of the wash steps to see that they were being interpreted differently between the sites, and that optimizing those steps in the protocol would help them with cell recovery. And so we resent new aliquots, got much better cell recovery from those sites. And now you see, decent recovery across all sites for both the PBMCs in red and then the spiked in so-called varicels that are the tantalum labeled lyophilized controls. So now we could really analyze the data. We took advantage of Visney plots, which show you a 2-dimensional representation of the high dimensional data and allow you to color by different markers. So here you see the major lineage subsets colored for each individual sample across the 4 sites. And this is comparing in A, the pre-stained samples that were provided by Mount Sinai versus the site-stained samples, which are really very similar all the way across. About the only thing you can really note here is that there was a little bit of loss of one of these samples here on the pre-stained side for site 3, but otherwise, and maybe a little bit over here as well, but that's really just a numbers issue. The patterns that were retained and the proportions are actually very, very similar. And we can show that more quantitatively. Here we're looking at frequencies of major lineages from B cells to NK cells across the 4 sites. So the little spark line here is showing you the 4 different sites as a separate line. And then you see below the intra and inter-site CVs. So if these lines are basically very flat and parallel, then you will have very low CVs over here and over here in terms of inter and intra-site. And if there's a bit more bumpiness and variability, then these CVs are a little higher. But what you can see, this again broken out by pre-stained on the left and site-stained on the right, is you can see really actually very low CVs, under 5% for intra-site and really under 10% for inter-site CVs, going up just a little bit in terms of the smaller population of NK cells for the site stain, but in general also still around 5% for intrasite and 10% or less for intersite, with this one exception. So that's analysis of the spiked in varicels, which were again the sort of control population in all of this, but we can of course also look at the PBMCs. Now you will see the 4 lines are diverging from each other a bit because these are 4 different donors. We expect them to have somewhat different frequencies of the different populations. And we tested here even more populations because these cryopreserved PBMCs could be stained with many more markers than what showed up well for the varicels. So again, though, we're looking for essentially parallel lines here that will give us then low CVs across the sites. We're not looking at intra-site CV in this case, again, because we know that there should be differences between the healthy donors, but we are looking at inter-site CV. And it's again coming in at around 10% or less for pre-stain and just a little bit higher for certain subsets. Here, basophils were an outlier, and they were a pretty rare subset, rather sometimes tricky to gate. And it was really just a couple of the sites that had some issues with that population, but generally, again, very low inter-site CVs. So finally, we did, with the help of MD Anderson and their statistics group, we did a variance component analysis, which looked at the degree of variation that can be attributed to either the site or the PBMC donor, meaning the subject, and then whatever error is still unaccounted for, unknown, is also calculated. So what you see here, again, for the pre-stained varicels, is you see that the vast majority of the variance in the middle portion of each of these bars is due to the subject. Sorry, this is not the varicels, this is the PBMCs of individual donors. And that's what you would want. You would want the variance to be due to the donor and not due to the site or something else in the protocol. If we look at the site stained, it's again similar. The biggest bar is always for subject. So most of the variance is coming from the subject and not from the assay itself or from the site specific variables. Okay, so that brings us to the conclusion. We felt that we could standardize quite well our CyTOF assays across sites, at least equivalent to what's been achieved with conventional flow cytometry, which is a standardization efforts that go back a number of years that I've also been involved with. And I can tell you that these CVs that I showed you here are at least as good and often better than what we saw with conventional flow. The use of the lyophilized or frozen cocktails is highly recommended based on our experience here versus in the first Gates-funded study. And also the addition, as I mentioned, of CD45 to the staining cocktail is a good step, even though you're using cryopreserved PBMCs where all of the cells really should be CD45 positive, but it gives you a stable denominator for calculating cell frequencies, and that led to better concordance across sites. The use of a labeled PBMC control that can be spiked in is a good idea. We used these lyophilized varicels in this work and have used them since in the pilot studies. And now as we're transitioning, we're actually going to be making our own PBMC controls that will be cryopreserved and metal labeled. So it's kind of the best of both worlds. Those are being pursued by the Dana-Farber for the use of the network. And we will make those with both a mixture of unstimulated and PMI ionomycin stimulated cells so that we have the ability to evaluate some of these activation markers that would otherwise be hard to see on resting cells. So that's all important lessons coming out of this work. And the final one, which is kind of a duh, it's obvious, but the precision is really dependent on acquiring sufficient events. And we learned that with the first round of this study. So the last thing I just want to mention is that we are also moving in the CIMAC network towards the use of a standard lyophilized cocktail across all sites. For the pilot studies that we did, we had site-specific cocktails and trial-specific cocktails, depending on the needs and desires of the study. But we're now going to be using this 30 marker panel that is sold by Fluidigm. They tell you there are 14 open channels, but really 7 of those channels are these cadmiums that are not particularly useful for most of the immunophenotyping markers. In fact, they don't sell them for purposes other than live cell barcoding. So in effect, it's really 7 open channels, and we actually have Fluidigm has, and we will be adopting a sort of default expansion panel of 7 markers that add a lot of interesting markers for immuno-oncology, including exhaustion and co-stimulatory markers. So this is not required. There may be cases where different expansion panels are used for the purposes of the study, but what we are going to be doing for sure is using this 30 marker backbone going forward in all of the CyTOF studies that we'll be doing across the network. So with that, I would like to stop and thank all of the people across the 4 CIMAC sites that worked on this project, and in particular, Adib at Mount Sinai for leading all of the analysis efforts, with Jack Lee at MD Anderson doing the stats that I showed, and the support of the NCI as shown here. And thanks to Pact also for supporting these efforts. And I'll be happy to take questions too.
Thanks, Holden. And so now I will introduce Dr. Sasha Nowchik, who will be talking about not only our multiplex IHC and IF, but a little bit about some of our other tier 1 and tier 2 assays. Sasha?
Very much, Dr. Adam. Thanks for asking me to present today about the harmonization efforts for multiplex immunohistochemistry and immunofluorescence. So I have some disclosures of research support, and I will discuss an immunohistochemistry method that we have a patent on at Mount Sinai. So the reasoning of our network is to establish biomarkers that can predict outcome to immunotherapies. And the most established ones so far for immune checkpoints have been tumor mutation burden. We heard about whole exome sequencing, as well as PD-L1 expression, which is done by immunohistochemistry. But we should also think of other potential predictive markers that are not yet necessarily FDA approved, but that have been there for a long time. For example, the first paper back in 1996 already showed that if you look at the tumor site at immunohistochemistry and the presence of lymphocytes in the tumor, you can rank them and see that there's heterogeneity among patients, some patients with very brisk infiltration, others absent. And this actually correlates with outcome, overall survival. This is true in melanoma. It's been also shown for ovarian tumors and for colorectal cancer. And it's probably best known now as the Immunoscore, which was truly established as part of the colorectal cancer prognostic effort, where quantifying cells such as T cells, CD3 or memory T cells or CD8 T cells, both inside the tumor and at its invasive margin, was able to cluster patients who will respond better than stage alone as information. So the idea of using immune cells in the tissue and measuring them has been there for a long time, and to find out if it's associated with a good outcome. But more importantly for our purposes, it's also noted that these immune infiltration are associated with not just general prognostic value, but also predictive value to a specific immunotherapy. For example, here is one of the first studies in Nature with using of PD-1 reagent pembrolizumab in advanced melanoma patients, where patients who had the clinical response also happened to have more T cells present in their tumor as well as the invasive margin, and then they went up with treatment, while the progressives seem to be more cold tumors. And more recently, it's important to note that it's not just the number of these cells that are important to the tumor site, but also where they're localized in relation to the tumor tissue and how they're organized. For example, this study here in bladder cancer shows that it's not enough to have T cells within the tumor site. They also have to go within the tumor nest themselves. If they're just consigned to the stromal areas, they're basically not having as good of a predictive value. So you have to have this combination of CD8 high with a stromal low signature with more of the T cells within the tumor nest. I mentioned PD-L1 as a marker, also that's very well established to help predict patients who may benefit more from immune checkpoints. Though it's important to know that it's just not PD-L1 generally expressed in the tumor site, it's also where exactly on the tumor site. Is it on the tumor cells or is it on the immune cells? And you can see that if you have both, you have the highest likelihood of response to treatment, while if you have only immune cells or only tumor cells, it's a little bit lower, and the worst being having no PD-L1 at all. But then again, I'm mostly talking about T cells and things like PD-L1, but also there are many other cells that could be of importance in terms of predicting outcomes, such as B cells and how they're organized sometimes as part of tertiary lymphoid structure or TLS. And you can see that here, these patients with melanoma or renal cell carcinoma treated with immune checkpoints, responders with the blue bars were higher in patients who had these either high density of B cells or of these TLS structures. So organization of these type of structures and quantification cannot really be done easily with single markers in immunohistochemistry, because you would end up using too much tissue. And this is why we all proposed as part of our CIMACs to introduce multiplex immunostaining as part of our protocol to be able to see many markers at a time with a sample sparing method. And so I want to immediately acknowledge the people who were involved in this harmonization effort, and most particularly Michael Tetzlaff, who is really leading all of this harmonization for immunohistochemistry at MD Anderson, who's now left but continues to work with our network. Edwin Parra there as well, Jack Lee as the statistician. Guray Akhturk was a pathologist in our group who led all our efforts at Sinai, and Evisa, Jeannie, and Anna Lacco, as well as Dana Neuberg for statistics in the group of Scott Rode, have participated in our Farber. So at first, we harmonized between these three centers because we had well-established methodologies, but we also had help from the NIH for our analysis, as well as Stanford that has another methodology for multiplex immunohistochemistry that was a little bit more newer and was a bit more complex to implement as part of our original pipeline, and I'll mention it briefly, the MIBI, but they were involved as well. And our goal was basically to compare the methodologies at each one of the three centers with the idea of achieving this relative harmony, which means that we don't necessarily need to agree exactly on the number of cells that are quantified in each tissue, as long as we agree what is high, what is low in terms of relative density of these immune cells in the tissues and different regions. The main issue, though, that we encountered very early on is how do we do this harmonization, given that each of our sites had very different platform for staining, as well as different protocols, and we were all already established when we proposed our assays as part of the CIMAC. So it was quite difficult to now come together and say, how will we be comparing our different platforms? And so very early on, we decided that in contrast to some of the previous harmonization efforts where we tried to really look at SOPs in them and streamline them to be as equal as possible, here we decided to harmonize our interpretation of the data rather than the SOPs themselves. And the reason for this is because we have different methods at each site, as I mentioned. So the MD Anderson site and Dana-Farber Cancer Institute site are using this Vectra system from PerkinElmer, now known as Akoya, which is using slides from formalin-fixed paraffin-embedded tissues, performing antigen retrieval and blocking steps, and then doing these consecutive cumulative staining of cycles using tyramide-based immunofluorescence dyes. And you can repeat this up to 5 or 6 cycles where you use different reagents for immune cells, and it adds on the colors and detection of these cells in the tissues. So it's a good method because it's been pretty well established in the literature, in particular in the field of immuno-oncology. Many people are using it. It has the advantage of offering the optimized antigen retrieval for each step and has high sensitivity. It has some drawbacks, such as the potential for immunofluorescence and limits of number of targets that can be used at once, because there is this fluorescence spectral overlap that you have to deconvolute as part of the methodology. And so far, it's been mostly consigned to small regions of interest in the analysis because of the computational power it takes to do these type of analysis. At Mount Sinai, we had a very different methodology that was really not at all immunofluorescence-based, but rather chromogen-based, and we call it multiplex immunohistochemical staining on a single slide, and or mix for short. And so it's the same idea of a general pathology staining where we use FFPE tissue and perform single stain chromogenic staining with an antibody, a secondary, and a chromogen, and we take an image of this particular staining. But then the new steps are that we perform this chemical destaining and then a blocking step, and can repeat this several times over and over, with the idea of having, for example, here CD20 destaining and then CD3, etc. So here we're not accumulating the markers, but we're doing them consecutively and taking an image after each one of our steps. So the pros are that we have the morphological features of the tissue that are intact because it's chromogen with counterstained with hematoxylin. So it gives you a lot of nice detail about the cell. It has high sensitivity. It can be done in a whole image, which is quite nice. And it's easy to implement because you really don't need much instrumentation other than a slide scanner. And if you want to do it for the staining itself, auto stainer. The drawbacks, again, is that it's a bit slow throughput because you have to do these consecutive stainings. The images that we generate need to be registered together if you want to look at co-expression of different markers. And eventually we do incur tissue damage, but we're still able to do now routinely about 10 to 12 markers on a tissue. So the main challenge has been, here is an example of our method for these markers like CD3, pancytokeratin, etc. Well, this is how the data looks in the MD Anderson and Dana-Farber Vectra platforms. But how do we segment cells, and how do we decide how many cells should be detected in the tissue, and then decide on which cell we call positive for specific markers? And again, here we had differences. We used QPath software at Mount Sinai, at the time at least of this analysis, and Inform software, which comes with the instrumentation at the other two sites. So to be able to do this comparison, we decided that we should really do it in a consecutive series of steps. And the first one was to align our protocols for scoring and quantification. And to do so, we, Dana-Farber, used a pre-stained set of images from both immunofluorescence and immunohistochemistry, and we shared these images across all the sites to compare our image algorithms, and also to compare the acquisition of the images from other sites as well, eventually by taking the original slides that were stained and sending them to the tumor tissue. So these two tumor tissues were sent again to each site as to be able to agree on the consensus panel for staining that included nuclear marker, cytoplasmic, as well as membrane marker, and then try to minimize our variables as much as we can to adjust our algorithms once again and cross-check the results with the other sites. And once we had this done, we went into the full validation, now using a tissue microarray with a variety of different tumor types, and to try to integrate each one of our protocols and compare our performance, and to still try to identify what contributes to variation in general, despite all these alignments. So this is a continuous review step that goes from the SOP sharing all the way to proficiency testing, which we'll continue to do to this day. So in the first step was harmonizing the IHC scoring algorithms. So we sent these pre-stained slides and selected 5 regions of interest at each site, and then asked the sites to quantify them the way they would normally do it, and compared it, and eventually adjusted how much segmentation should be happening, whether we were over-segmenting some cells, and whether some of the positivity calling was maybe too sensitive or not sensitive enough in some sites. And when we changed this along with other quantification for the markers labeled here on a series of different pre-stained tissues. So we selected them on purpose for nuclear staining, cell membrane staining, as well as more cytoplasmic staining. But we were able to achieve a fairly good concordance. So we thought that at least in terms of algorithm, despite the difference between Inform and QPath, we were able to agree pretty much on how we would quantify these tissues. So the next step was to do the entire staining protocol harmonization, which was much more challenging, as I mentioned, because of all the variables that we had. So the table here shows in black the only things that were common to each site, and that was the platform for staining, the LICA bond, as well as our CD8 clone and our cytokeratin clone. But for the other markers, we had different clones used. So some of them were shared by 2 sites in green, or some of them were unique to just 1 site in red. And we also went on and stained for these 2 head and neck tumor samples. And after we aligned our protocol once again and looked at our readout after staining, so I'll skip all these steps, we found out that there were a couple of lessons that could be learned. And one of them, that despite trying to get the same tissues from the same tumor block, we could see that some of the sites received areas of the tumor that were actually quite different in the specific matched region of interest that we decided to select. And partially, one of the reasons for that was that we didn't necessarily recut a fresh slide at one of the sites. And in the end, the slides that we obtained in 2 others were a little bit further away from the original block. And that introduced obviously some differences in the quantification that were not due to the method itself, but more to the tissue. However, in the method, we also realized things like PD1, for example, at Mount Sinai, seemed to be underperforming, and we always had consistently less staining compared to the other 2 sites. And we realized by comparing just these tissues that our clone was not that great, and we decided to switch to the one from MD Anderson. And so once we've thought out now for the next stage that we had to make sure that our slides that we analyze are truly consecutive and that we use the best clones we can at each site, we decided to go on with the tissue microarray. And so this was assembled at DFCI with a variety of different tumor types within 1 slide that were basically cores of a few 1 millimeter wide, if I remember correctly. Each one were stained at each site respectively and acquired with the images and generated all the data. And then with all 3 of us, we sat together and selected within each one of those tissue microarray dots a little area region of interest that was then analyzed and quantified. And we adjusted again 1 more time the algorithms if necessary, or restaining if needed in case of the PD1, and then went over to the harmonized data. And so this is the end result of this analysis, where here are the stained examples of these microarrays, and we stained them for 5 markers labeled here: CD3, CD8, PD1, PD-L1, and cytokeratin. And you can see in the boxes, the box plots here represent each one of the tumor types present on these arrays: bladder in red, head and neck here, lung, melanoma, and renal cancer, where we had each 4 to 5 different tumors from each tumor type. And then the way that you read these charts is looking at the correlation that we found across sites. So in a 1-to-1 comparison between Sinai and Dana-Farber, Sinai and MD Anderson, and Sinai and Dana-Farber and MD Anderson. And if the comparison is good, you should expect this diagonal, where for each one of the tissues, you have a very good concordance in the quantification of these cells. And really, as you can see, remarkably, we found out that all of these markers ended up to be truly nicely concordant, not just in their relative abundance in each tissue, but actually even in the percentile and amount of cells per millimeter square that were found in these particular regions of interest. There were only a few outliers like PD1 here that could still be ascribed to mostly, or PD-L1 that could be ascribed to mostly tissue differences in the region of interest, despite the fact that there were consecutive slides. And overall, we had preset this acceptance criteria for a correlation that should be higher than 0.7, and it achieved for all the markers, with cytokeratin being maybe the one that's the most difficult because it's so dense and difficult to actually quantify precisely because it's on every single tumor cell. And the coefficient of variation was preset as 0.4 as our acceptance criteria, and only a few of the samples were a little higher than that, but on average it was less than 10% of coefficient of variation. So overall, we were very happy to see this comparable quantification interpretation of our result, despite these different instrument platforms, clones, operators. And we're currently still continuing to align our methods as we go on to use now these multiplex staining platforms for our trial samples. And the goal will be to not just be confident that each of our method is reliable and compares with each other, but allow eventually for cross-trial analysis as well, so that we can try to think, is there something in general in checkpoint blockade across different tumor types that we can think about in terms of immune infiltration changes. So this is part, as Stacy was mentioning in the introduction, of a series of assays that we prioritize as tier 1, being recommended to be done on all trials and chosen for their balance between reproducibility and innovation. And we've heard a little bit about some of the other trials, but I just want to touch briefly in the last few minutes that I have on some of the other assays that we're performing as part of our network, starting with some that are not yet considered tier 1 because they're a little bit more experimental, but maybe, and this technology that we're all very excited about, such as spatial transcriptomics, and then mention Olink and microbiome as well. So Stanford is the site that uses MIBI as part of their multiplex immunochemistry offering, and MIBI is pretty much the similar idea like site of using these metal conjugated antibodies that you can put on a tissue resection slide and put about 36 at a time on these tissues to be able to then be quantified by this mass spectrometry, where you generate this ion beam on the tissue and shave off a few microns or even less, so a few nanometers, 100 nanometers from the surface of these cells, and be able to quantify the markers and look at them. And so this is a paper that the Stanford group produced, showing that they can, in this breast cancer tissues, see a variety now of different immune cells. And so right now we're in discussions. They've been generating the validation document to make sure that this can be used now as part of a clinical trial. It's a little bit slower throughput and more complicated to implement because of pre-analytical variables that are required for these tissues to be analyzed. But we're thinking that by the use of reference materials that we have now generated for our future of our network, where we'll be comparing continuously our methodologies and how they perform, Stanford could be part of these comparisons together with the other 3 CIMACs to be able to align our protocol. We're also very interested in going into further methodologies for the analysis of tissues, and one of the hot topics this year has been spatial transcriptomics, and it's a very in development technology which will probably change a lot in the next coming year. But there are 2 platforms that have been looked at the most probably, which includes the NanoString GeoMX platform as well as the 10x Genomics Visium platform, and we're currently doing some some QC for looking at how these methods compare to each other, as well as what type of pre-analytical variables are required to make sure that these tissues are well analyzed. And so far, there are drawbacks for these methodologies, that they're not yet at the single cell resolution level, but eventually the idea of getting the information from both proteomic markers as well as transcriptomic markers would be quite nice to be able to cast a wide net of understanding in the most unbiased fashion possible which cells at the tumor site change with immune oncology treatment. The other assays that we're very excited about, and that you actually heard from Kathy before, was the Olink assay. So this is the platform that was chosen as part of our CIMACs for analyzing soluble analytes from plasma or serum. And it allows you to quantify inflammation and chemotaxis markers with the idea that these circulating proteins in the blood could potentially reflect local tumor events, such as secreted or cleaved proteins and molecules shed through exosomes, for example. And you can do that repeatedly, since it's easy to obtain blood, and it's very sample sparing. Just a microliter of serum is enough for this assay. And you can use 92 analytes in once and to correlate them with clinical outcome of treatment. So we chose this Immuno Oncology panel from Olink, which includes a lot of activation markers for T cells, for example, chemokine and chemotaxis markers, suppressive markers potentially of tumor immunity, vasculature markers, as well as tumor apoptotic markers, for example. And we've put that, the way that it works is basically looking at each one of those 92 proteins with a pair of antibodies. And if the protein is detected through both antibodies binding to the protein, they will anneal through these DNA sequences that are linked to the antibodies, and you can then extend them by PCR. So it's a very high dynamic range and sensitivity because of this PCR-based detection. And we've analyzed so far over 1,400 specimens across 8 different trials and 445 unique patients. And it's been performed at a single site at Sinai, so that facilitates our approach because we don't have to harmonize yet, but it will make it quite easy to do cross-trial comparison because the platform is very stable and always the same panel used throughout every study. And this is an example of an application for CAR T-cell treated myeloma patients, for example, where you can see that not all patients who develop these cytokine storms after CAR T-cells have the same type of cytokines that are produced. Some of them have these type of chemokines, while others have a different subset. So you can maybe have a bit more granularity as well about detection of which cytokines could be pathogenic and associated with adverse events here of treatment with immunotherapy. And finally, one of the other assays that we're very interested in developing has been the sequencing of the microbiome, because there's been a bunch of papers showing that the gut composition of the microbiome could be very important in supporting immune responses as part of the immune checkpoint treatment. And to be able to consistently look at what are those bacterial strains present in tissue stools, in stool of the patients, we've done efforts to make sure that our methods are, again, reproducible and comparable across different sites. And one of the ways that we do that is with the use of these mock communities or Cymac mock communities. We can do some spike-ins as well and reference controls and see that in general, their batches are very reproducible from and unique to each patient and cluster well together. So these are SOPs that are all present in the website that Ignacio mentioned. And with these longitudinal reference controls, we hope to be able to continuously also compare our different platforms here between MD Anderson and Mount Sinai as the 2 sites participating. Eventually, when we get all this data, we need to also figure out how to interpret it. And we're also thinking about analytical pipelines within each trial, but also across trials, and take all the data generated from our various tier 1 assays, put them together, and include things like clinical measurements as well and outcomes. So you can apply a bunch of linear mixed models and other statistics, as well as visualization tools such as PCA and UMaps, to be able to interpret our results and hopefully get data that is useful for identifying these biomarkers. So our goal is to now integrate these methodologies as much as we can into a set of both tissue level information, but also from blood and host level information. Also go more on automation and potentially machine learning. So we're currently in the process of automating all our analytical pipeline from QPath to something that's maybe running more automatically with algorithms that are part of a general pipeline. And this will allow us to go faster into our analysis, and we'll have to still make sure that it's comparable to what we used to do before. And the eventual goal is to combine those biomarkers together. So we know PD-L1 and TMB as 2 of the well-established ones for immune checkpoints, but we want to introduce things like these infiltrating lymphocytes, but potentially all the other assays that we've mentioned, such as microbiome, cytoph information, maybe circulating tumor DNA. And the idea is that if we combine those together, even though each one of them is really imperfect to define outcome to immunotherapy, together they may be stronger as a composite biomarker. So to conclude, I think we learned here that we were successful in our harmonization of multiple different methodologies. And interestingly, you don't necessarily have to do to enforce an SOP across the different sites to be able to achieve this, but allowed to be flexible, and you have to be ready to change some protocol steps when needed. It's important to have this continuous assessment of performance with every protocol change, and that's why we develop reference materials in the future to do so. The goal is to allow to compare, to allow our methods to evolve, because we don't want to be stuck into one assay for the next 5 years and still be able to compare it over time across studies. And pre-analytical variables are probably very critical to all our work, and sample heterogeneity and patient heterogeneity is still usually the dominant source of variation in our data. So in general, as known for other previous protocols for harmonization, one of the most important factors is also to have a well-established protocol and operator at the individual sites that tends to be more reliable to obtain reproducible data. And so I want to thank again the people involved in this work, in particular the people, Gurai at Mount Sinai, as well as Sung-Hee Kim Schultz, which leads all the OLINK efforts, and Edgar, our computational efforts, the team at MD Anderson and Dana Farber, who contributed a lot, as well as our efforts here, our collaborators from FNIH, NCI, and Stanford, and the support from our grants. Thank you.
Thanks, Sasha. If you'd like further information on these assays, we'd be happy to follow up with you and speak about it later. And in addition, as I promised, we wanted to briefly just refresh everyone's memory. We are still accepting applications for trials to partner with the Pact side of the Pact CIMAC CIDC network. We are still reviewing applications. We'll be accepting those until June 30th of 2022, with the need for all sample accrual to happen no later of a drop dead date of January 31st, 2023. So we appreciate all of your time and efforts in joining us today, and we look forward to hearing from you if you would like to partner with us in the future. Thanks again to our speakers, Sasha, Holden, Kathy, and Ignacio. Very much appreciated. Thank you, everyone, and have a great day.